Hello students. Today one more talk about loose stool. One more tutorial class as a revision to you. So today we'll be talking about watery diarrhea. In my previous tutorial class I have taught you bacillary dysentery. It is somewhat the same, but the difference lies in the fact unless and until the complication turns up, there is not involvement of blood and mucus here. So it's just loose stool, rather watery diarrhea. So first let's begin with the definition of what diarrhea is. Talking about it, there are certain terminology you must mention. Firstly, there must be 3 or more loose or liquid stools per day, and it would last for several hours or days. Now what it can be caused by? It can be caused due to microbial infection or maybe certain sort of gastrointestinal diseases. Now we can classify watery diarrhea into 3 components. One, acute, which will last for less than 2 weeks. Second one being persistent, which is continuing for 2 to 4 weeks. The third being chronic, which is going beyond 4 weeks. Now, who are the etiological agents by whom it can be caused? Firstly, it's the inflammation of the small bowel. For bacillary dysentery, I talked about it's the distal ileum and colon. This one, watery diarrhea, it involves the entire small bowel, and the transmission is same, fecal-oral route and contaminated food and water. The bacterial causes are mainly due to the enterotoxin which is secreted, mainly Vibrio cholera, enteropathogenic, enterotoxigenic, enteroaggregative E. coli, Clostridium perfringens, and etc. There can also be viral causes, and the most important one being rotavirus. Apart from that, parasitic causes can also be there, Giardia lamblia, Cryptosporidium species, etc. So the chart is given right in front of you, you visualize it and memorize what can be the etiological agents involved. Moving on to the pathogenesis, the first and foremost fact is that you have to identify to what extent the etiological agent must be involved to manifest watery diarrhea. So what is the meaning of the word inoculum size? It is the infective dose, that is minimum dose required to initiate the infection. Now depending upon various species it differs. For example, say Shigella, enterohemorrhagic E. coli, for them it's just 10 to 100 bacteria or cyst. But in case of species like Salmonella, it's 10 to the power 3 to 10 to the power 5 bacilli. In case of Vibrio cholerae, it's 10 to the power 5 to 10 to the power 8 bacilli. So depending upon the species, the infective dose or the inoculum size differs. Next, once the bacteria or the virus or the parasite has invaded, or rather it is in a sufficient quantity, it will have the tendency to get itself attached onto the host's body. So that is what you call as adherence. Now it will adhere with the help of special adherence structures which are present in the particular etiological agent. Now it does so by competing with the normal bowel flora and thereby colonizing in the intestinal mucosa. After it has adhered itself onto the host's body, after the immune system does its work, it will have the tendency to multiply, and definitely toxin production is manifested. I have already said the enterotoxins are mostly involved if it's a bacterial invasion, and thereby toxin production will do the rest of the work. There are certain host factors which may also trigger the manifestation of watery diarrhea. What can they be? They can be suppression of normal flora, neutralization of gastric acidity, which can also be done due to some medications as well, inhibition of intestinal motility, impaired host immunity, etc. So these factors are well known to you because I have already talked about such host factors previously when I was teaching you bacillary dysentery. Moving on to the clinical manifestations, as in what diseases can it cause. Obviously there will be loose watery stool, 3 or more passages rather. Abdominal cramps can also be there, weight loss, vomiting, nausea, fever, etc. Now lab diagnosis, just the same. For bacillary dysentery, we took a fecal specimen and we contained it in a fecal specimen container. The same is applied over here. It's the sterile screw-capped wide-mouthed container in which the fecal specimen is collected, and it should be done with utmost sterility. and you will transport it in transport medium. In case of bacillary dysentery, I talked about Sachs buffered glycerol broth or gram-negative broth. Here we use Venkataraman Ramakrishnan medium or Carey Blair medium. So these words are well known to you because you have already studied Vibrio, so this transport medium are well known to you. Moving on to the microscopy, the first and foremost thing that you perform is wet mount. Just simply you take one drop of stool and you put it on a clean glass slide, and after that you cover it up with a cover slip. So you observe certain characteristic features. What can they be? These can be detection of pus cells, parasitic cyst, trophozoite, eggs or larva. See, whatever is the infective stage of that particular parasite, that's what you will identify in the stool. So depending upon that, you will identify cyst, trophozoite or whatever it is. So as you study parasitology, you will get to know it. So you prepare a saline or iodine mount. It's the wet mount preparation. Directly it can be done, or else you put one drop of saline, or else you put one drop of iodine onto which you add stool in it. Here is a video showing the darting motility of Vibrio cholerae. In this particular device it's not playing, but don't worry, when I give you the PPT you will get the video out there. Otherwise you can also Google it, the darting motility of Vibrio cholerae. It's a beautiful scenario to observe. Anyways, so moving on to the gram stain smear, if you presume it to be a Vibrio cholerae, then you will find the comma-shaped bacilli. You can see in the figure comma-shaped bacilli out there. If you perform acid-fast staining and you are suspecting the species can be Cryptosporidium isospora, then in such cases you will observe oocyst after doing acid-fast staining. Electron microscopy can also be performed. For example, if you find rotavirus, you will find spokes are grouped around a hub of wheel. So a wheel and around it the spokes are coming off. So as you study the viruses or rather viral diarrhea, these things you will be getting to know. So it's just a summed up thing, okay? Now you perform the bacterial culture. This thing I have taught you previously. You need an enrichment medium, you need a mildly selective media, and the last one is highly selective medium. So if it's an enrichment broth, you can ask for selenite F broth or alkaline peptone water or tetrathionate broth. For mildly selective media, it's the McConkey agar, highly selective media, DCA, XLD and TCBS. After the bacterial culturing is done, you perform biochemical tests, serotyping and of course AST, antimicrobial susceptibility testing. See, this is the figure, the same thing I have taught previously as well. These water, the color of the consistency of this particular broth medium will become turbid, which will indicate that there is the growth of bacteria in it. So selenite F broth, basically it's for the isolation of Salmonella Shigella. Tetrathionate broth, as you can see, it will not allow the growth of regular normal coliforms, and alkaline peptone water, it is specific for the enrichment of Vibrio cholerae. Now we move on to the mildly selective media, mainly McConkey agar. Look, in case of McConkey agar, I am repeating it once again, this is a differential media. That means it will help you to differentiate between lactose fermenting colonies and non-lactose fermenting colonies. The figure on the left hand side is the lactose fermenting colonies and the right hand side one is non-lactose fermenting colonies. In case of McConkey agar, there is presence of bile salt and there is presence of crystal violet. These two stuff will not allow the growth of any gram-positive organism or any, say for example, fastidious gram-negative organisms will not grow. They will be inhibited from being grown due to crystal violet and bile salt. Besides, lactose is the source of carbohydrate here and neutral red is the indicator. What is the function of neutral red over here? The color changes that you can see. Somebody is pink, somebody is not pink, somebody is pale. So these color changes are happening because of the change in the acid. That changes are being done by the indicator, neutral red. So next is the highly selective media, the first one being DCA, stands for deoxycholate citrate agar. So deoxycholate citrate agar, talking about it, this is also a differential media which will help you to identify the organisms which are able to ferment the carbohydrate and the ones who are not. Same, lactose is again the source of carbohydrate over here. Besides it being a differential medium, it's also selective because it will be allowing the growth of specific enteric pathogen, namely Salmonella and Shigella. So deoxycholate salt and citrate, these two things will not again allow the growth of gram-positive organisms, and the normal commensals which are there present in our intestine, they will also not grow. You see a difference over here. The left hand side diagram has got black centered colonies and the right hand side diagrams does not have so. Why is it so? The left hand side one is Salmonella, which is shown, but in here it is having black centered colonies because over here ferric ammonium citrate is utilized, or rather it is converted into iron sulfide. Such a change, where does the ferric ammonium citrate come from? This came from the medium. So if the bacteria is able to convert it into iron sulfide, that means the bacteria has got the ability to produce H2S. So to identify the production of H2S, we do it so, and we can see it with the black centered colonies. From it we can clearly understand that Salmonella is somebody who can produce H2S, but the pale colonies who are not showing any black center are not the ones who can produce H2S, for example Shigella. Moving on to the next one, XLD, xylose lysine deoxycholate agar. So here again we are having deoxycholate, so of course it will have the function just like that of the previous one. Here we are using 3 carbohydrates. What are they? Lactose, sucrose and xylose. Now the organisms who are able to ferment or utilize more than 1 carbohydrates, then they will be showing yellow colored colonies. That means they are basically lactose fermenters, for example E. coli. Moving on to the other one, who are not able to utilize any of the carbohydrates, they will show translucent colonies. You see another point of difference over here. Here the left hand side again shows black centered colonies, the right hand side shows again the translucent colonies. So somebody is showing black centered colonies. So the concept lies the same over here. Sodium thiosulfate is used as the source of sulfur. Ferric ammonium citrate is converted into iron sulfide, which indicates that the bacteria is able to produce H2S. This is typical for Salmonella, not for Shigella. So by this you can identify where is Salmonella growing and in which cases Shigella is growing. Besides, the indicator over here is phenol red. The previous one, DCA, the indicator was neutral red. XLD, the indicator is phenol red. Moving on to another highly selective media, it is TCBS, stands for thiosulfate, citrate, bile salt, sucrose, agar. So TCBS is selective for Vibrio species. So mainly it will identify the Vibrio species out over here. You see in the left hand side, yellow colored colonies are there. These are sucrose fermenting Vibrios. So sucrose fermenting Vibrios, for example Vibrio cholerae, it is showing yellow colored colonies. The non-sucrose fermenting Vibrios, they will show green colored colonies. Next is tissue culture. I have already said E. coli is a normal flora that you find in stool, but I am also talking about E. coli, certain species of which are causative agent, are etiological agents of watery diarrhea. So how do you identify them? Already I have said you can do serological test in that. Apart from that, you can also perform tissue culture, which will help you identify the enteric viruses and differentiate it from some of the diarrheagenic E. coli. For example, ETAC, it can penetrate HeLa and Hep2 cell line. But in case of EHEC, it is detected by the cytotoxic effect on the varicose line. So, observing such phenomena on tissue culture, you can identify whether that E. coli is a normal commensal or it is that hemorrhagic species or toxigenic species and aggregative species or not. Antigen can also be detected. For that you require ELISA, and rapid test can also be performed. Rapid test as in latex agglutination test, wherein you will identify the antigens. Remember, the enterotoxins which are involved, they also have got some antigenic properties in them. So by identifying the toxin which are mediated by the particular bacteria, you can also perform the antigenic detection. Molecular method, PCR, is always preferred, the gold standard method. Toxin detection can also be done using ELISA, immunodiffusion, neutralization, and LAT for detection of enterotoxin in the stool. Now treatment. I have classified this treatment methodology into 3 types. One being mild diarrhea, another being moderate diarrhea, and the last being severe diarrhea. What are they? Talking about mild diarrhea, it's just 1 to 2 uniform stools per day. Moderate is 3 to 5 uniform stools per day, and severe diarrhea is greater than equal to 6 uniform stools per day. Besides, there are other symptoms too. The table is given to the treatment that should be provided for such cases, so observe it minutely. How can you prevent it? Of course, sanitization is definitely required. Hand sanitation is required. Hand washing should be done. Clean food must be intaken, and you have to take all precautions so that reservoirs can be eliminated and the channel of transmission can be well broken. And definitely, providing nutritious food can also help us in preventing watery diarrhea. So that was all about watery diarrhea. If you're having any sort of a doubt, definitely you can ask. There can be questions MCQ on the basis of this particular tutorial class. Thank you.